Welcome to this explainer on the continuity and variation section of the C-Sec biology syllabus. Today, we're taking a fascinating, really dynamic journey. We're going to start incredibly small, looking at the microscopic building blocks of life hidden deep inside our cells, and then zoom all the way out to witness the evolution and, well, the survival of entire species. By breaking this down step by step, we'll make these complex concepts super clear.
Okay, let's dive right into this. Here's our roadmap for today. Part 1 is the blueprint of life. Then, Part 2, cell division and growth. Number 3 covers inheritance and variation. Moving on to 4, we've got evolution and selection. And finally, Part 5 is genetic engineering. Let's tackle these one by one, shall we?
Section 1, the blueprint of life. To really understand how life continues, we first have to transition into the microscopic world. We need to look at our genetic makeup, which basically acts as this highly detailed instruction manual for building a living thing. So it all starts with DNA, right? Deoxyribonucleic acid. You can really think of DNA as the master instruction book of life. It's the nucleic acid that contains absolutely all the genetic information required to build and maintain an organism. Honestly, before we can figure out how traits are passed down to the next generation, we have to deeply understand this fundamental molecule. Now, what's really interesting about this is how that instruction book is organized. If DNA is the master book, then chromosomes are its chapters. They're made of DNA tightly packed together with histone proteins, and within those chapters, you have genes. These are specific segments of DNA that act kind of like individual recipes to produce a specific protein. And alleles, well, they're simply the different flavors or variations of that exact same recipe. We also have to track how these chromosomes are counted. Haploid, represented by N, means a cell has a single set of chromosomes. Diploid, or 2N, means it has paired sets.
Moving right along to Section 2, cell division and growth. You see, life only continues if this genetic code can actually be accurately copied and mixed.
There are two entirely different ways cells divide, and contrasting them is absolutely crucial. First up, mitosis. Think of this as the engine for physical growth and asexual reproduction. It's like when sugarcane cuttings grow into identical new plants. It's all about exact replication, maintaining that paired diploid or 2N chromosome number. Now, meiosis is a totally different ballgame. It purposefully halves the chromosome number down to a single haploid, or N, set. Why? To form gametes, like pollen, ovules, or sperm. And crucially, meiosis scrambles the genetic code through processes like crossing over. That is the vital engine of uniqueness and genetic variation necessary for sexual reproduction.
Mastering these definitions directly translates to success on your CC exams. Examiners will specifically test your ability to define terms like mitosis, artificial selection, and genetic engineering. They want you to articulate that mitosis maintains identical cells for growth and explain exactly why that matters. Nailing these core concepts for sure ensures you're perfectly prepared for these exact questions.
Section 3. Inheritance and variation. Okay, so we know meiosis creates unique traits, but how are those unique traits actually passed from parents to the next generation?
So the crucial point here is that variation isn't just about things looking pretty, it is literally nature's ultimate insurance policy. As the syllabus emphasizes, variation makes it far less likely that changing environmental conditions will wipe out an entire species. Think about the variety in your own backyard, the different colors of Carabinobicus flowers, or the unique patterns on local frogs. That diversity within a single species, that's their key to long-term survival.
When those traits are passed down, they follow some very specific rules. Some traits are dominant, while others, like albinism, are recessive. Sometimes traits kind of share the spotlight in what we call codominance, which is perfectly illustrated by human blood groups. You'll also encounter sex-linked diseases like hemophilia or colorblindness. To succeed here, you need to be entirely comfortable predicting how these traits mix. You'll use genetic diagrams like Punnett squares and pedigree charts. You have to know the difference between homozygous and heterozygous genotypes. That's the invisible genetic makeup, and the phenotype, which is the physical expression you actually see. Now, when these traits are physically expressed, they fall into two main categories. First, we have continuous variation. This involves traits that exist on a sliding scale with a whole range of intermediate values. Think of a measuring ruler. Your foot size or the size of a leaf falls somewhere on a spectrum. Discontinuous variation, on the other hand, is strictly either-or. Traits fall into distinct categories. It's like sorting files into strict folders. You can either roll your tongue or you can't. Cattle either have horns or they don't.
Section 4. Evolution and selection.
We're zooming out quite a bit now. We're looking past individual genes to see how these genetic variations in entire populations interact with the environment over generations. Let's walk through the actual mechanism of natural selection step by step. It starts with genetic variation existing as a template, often sparked by a random mutation. Next, the habitat throws down a challenge, some sort of selective force like a new predator or a shift in climate. From there, nature preserves the organisms with useful adaptations that help them survive those challenges. Finally, those survivors reproduce and the population retains these beneficial genes over time. We see this in the real world constantly. Think of bacterial resistance to antibiotics or how Caribbean lizards physically adapt to distinct island environments.
And you know, this abstract concept of evolution is deeply, urgently relevant right now. A teacher's note right in the CSEC syllabus states, "Our territories are faced with drastic changes due to human activity, overpopulation, limited resources, susceptibility to natural disasters. Small island developing states in the Caribbean are facing massive selective pressures. So understanding adaptation isn't just textbook theory. It's literally a matter of survival for local biodiversity."
Over long periods, this environmental selection leads to speciation, the formation of totally new, distinct species. This can happen through physical, geographic separation, like a new island forming, or it can happen through ecological and behavioral differences, like a change in courtship rituals. But we have to be careful here. If environments change too rapidly, species can't adapt in time, and they face extinction. Sadly, this is a tragic reality we've seen with the Caribbean monk seal due to habitat loss.
And this brilliantly illustrates the stark contrast between nature's way and our way. Natural selection is nature's blind survival test. It's driven entirely by environmental survival pressures to preserve useful adaptations. Artificial selection, on the other hand, that is human-directed. It's humans actively selecting and breeding specific traits in plants and animals to suit human needs, like breeding plumper pigs or higher-yield crops for agriculture.
All right, that brings us to Section 5, Genetic Engineering. We're stepping way beyond selectively breeding animals now and moving into directly manipulating the very code of life itself.
Genetic engineering basically involves inserting genetic material from a completely different organism. We are effectively cutting and pasting DNA. The modern applications here are just revolutionary. In medicine, we've engineered bacteria to produce human insulin. In agriculture, scientists inserted beta-carotene genes into rice to combat night blindness. The syllabus also expects you to be familiar with tissue culture and human cloning, creating entirely genetically identical individuals. It's wild stuff.
But this leaves us with a massive question to ponder. As we manipulate the very blueprint of life through cloning and genetic engineering, what are the social and ethical boundaries we must respect? It's a heavy responsibility. Well, we've journeyed all the way from the microscopic structure of DNA to the global engineering of our crops. I really hope this explainer has helped you master the continuity and variation of life. Keep questioning everything and stay curious.